In this part you will learn more about specifications. A specification is a contract between the authorities and the company regarding the quality of the product. In practice, it is a list of tests, references to analytical procedures, and appropriate acceptance criteria which are numerical limits, ranges, or other criteria for the tests described. It is a combination of demands set by authorities which are often found in pharmacopoeias and discoveries during product development. Important guidelines for specifications are the ICH Guideline Q6b. Specifications should be based on data obtained for lots used in preclinical and clinical studies. The quality of the material made at a commercial scale should be representative of the lots used in preclinical and clinical studies. Specifications do apply to all produced batches of active substances and the final product, but we also have specifications for the starting materials used to produce these products. The analytical methods used should be either pharmacopoeia methods, and these should be qualified before use, or methods that are validated during the development of the drug product. The tests carried out on biologics consist of a mixture of standard tests that are known to be necessary for a particular type of product, and product-specific tests that have been identified during the development of the product. These tests are conducted to ensure both safety and efficacy. In terms of safety and efficacy, it is important to verify that the product contains the correct active substance and has the appropriate attributes. Therefore, the identification test is conducted on all products. Efficacy also requires that the product has the correct content and uniformity of content, and thus this is also tested for all products. For biologics, a potency test is conducted as the structure, not just the content, is crucial for efficacy. There are also tests that primarily focus on safety, such as testing for impurities, endotoxins, foreign particles, and sterility. Additionally, there are a range of other tests that verify that the formulation is correct, such as pH levels and the content of preservatives. The key to a good specification is to conduct tests that are necessary for product safety, without testing anything that is unnecessary. A specification always contains a description of the appearance of the drug product, including if it's a liquid, dispersion, or a lyophilized powder, and also other information such as color, transparency, etc., that the product contains the right drug substance, is tested in the identity test. For biological drugs, this can be done by several different tests, where for example tests of biological activity and potency can be included. It is important that the test is highly specific for the drug substance and should be based on unique aspects of its molecular structure and or other specific properties. More than one test might be necessary. Assessment of the biological properties constitutes an equally essential step in establishing a complete characterization profile, a so-called potency test. An important property is the biological activity that describes the specific ability or capacity of a product to achieve a defined biological effect. A valid biological assay to measure the biological activity should be provided by the manufacturer. Examples of procedures used to measure biological activity include animal-based biological assays, which measure an organism's biological response to the product, cell culture-based biological assays, which measure biochemical or physiological responses at the cellular level. Biochemical assays measure biological activities such as enzymatic reaction rates or biological responses induced by immunological interactions. Other procedures such as ligands and receptor binding assays may be acceptable for some products. Drug products may have impurities due to manufacturing or storage processes. Qualitative and quantitative analysis is important to measure these impurities accurately. Pharmaceutical companies establish acceptance criteria to meet quality standards based on in-house research and regulatory requirements. Impurities include host cell proteins, DNA, cell culture-derived impurities, and downstream-derived impurities. Clearance studies may be used to eliminate the need for establishing acceptance criteria for certain of these impurities. Product-related impurities may result from the aging of the product and include chemical and physical degradation products. Impurities from chemical degradation can be detected and characterized by chromatography, such as HPLC-MS, or other relevant analytical methods. Aggregates and unwanted oligomers of the active substance can be analyzed using methods such as electrophoresis, size exclusion chromatography, and field flow fractionation. Injectables are always tested for sterility, endotoxins, and particles. 100% sterility is difficult to measure, and theoretically, it is defined as one non-sterile product per million. In reality, sterility testing is not allowed to fail. Thus, a product is out of specification if it fails sterility testing if there is no obvious reason to suspect analytical errors. Endotoxins and pyrogens are similar types of tests. The purpose is to make sure that the product does not have rests of dead bacteria that can trigger an inflammation-like response in the patient. Traditionally, pyrogens were used and they detected the presence of pyrogen compounds using animal testing. Today the tests are most commonly for endotoxins, which are lipopolysaccharides from gram-negative bacteria. The test used is the Limulus amoebocyte lysate test, LAL. Foreign particles and aggregates can be dangerous, as they can trigger inflammation responses, anti-drug antibodies, and clotting. All products are therefore tested for visual particles, while a few vials in a batch are also tested for subvisual particles according to USP 788, particulate matter in injections.